Section 0 of Gray's Anatomy, Part 1. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recorded by Laurie Anne Walden. Anatomy of the Human Body, Part 1, by Henry Gray. Preface to the 20th edition. Since the publication of the first English edition of this work in 1858, and the first American edition in 1859, great advances in the subject of anatomy have been made, especially in microscopic anatomy and the anatomy of the embryo. This knowledge was embodied from time to time in the successive editions until finally considerable portions of the text, sometimes sections, were devoted to these subjects. However, the main text has always remained primarily a descriptive anatomy of the human body. In the present edition, the special sections on embryology and histology have been distributed among the subjects under which they naturally belong. New matter on physiological anatomy, laws of bone architecture, the mechanics and variations of muscles have been added, occupying much of the space formerly devoted to the sections on applied anatomy. The sections on the ductless glands and the nervous system have been largely rewritten. In the latter, a more rational presentation of the sympathetic nervous system has been achieved through the use of diagrams and descriptions based on physiological and pharmacological work. The central connections of the spinal and cranial nerves are also emphasized. Illustrations have been added wherever important points could be made more clear, and throughout the work colored pictures have been even more extensively used than heretofore. In this respect, special mention might be made of the central nervous system and the section on the muscles. In the section on syndesmology, 6 illustrations are used from Quain's Anatomy through the courtesy of the publishers, Messrs. Longmans, Green & Company of London. The use of the BNA nomenclature in English has been retained practically unchanged in this edition, and important references to the literature have been added at the end of each section. As a practical work on the subject for the student, Gray's Anatomy has always been recognized and appreciated. The plan originally formulated, which has proved so successful, has been adhered to as much as possible. It is interesting to note that although Henry Gray saw only the first edition, much of the original text persists, and many of his illustrations are still in use. Bearing this in mind, it has been the endeavor of the editor to supply only such changes as advances in the science made necessary in order that this work may reflect the latest accessions to anatomical knowledge. W. H. L., Baltimore, 1918. Introduction. The term human anatomy comprises a consideration of the various structures which make up the human organism. In a restricted sense, it deals merely with the parts which form the fully developed individual and which can be rendered evident to the naked eye by various methods of dissection. Regarded from such a standpoint, it may be studied by 2 methods. 1. The various structures may be separately considered, systematic anatomy. Or, 2. The organs and tissues may be studied in relation to one another, topographical or regional anatomy. It is, however, of much advantage to add to the facts ascertained by naked eye dissection those obtained by the use of the microscope. This introduces 2 fields of investigation, that is, the study of the minute structure of the various component parts of the body, histology, and the study of the human organism in its immature condition, that is, the various stages of its intrauterine development from the fertilized ovum up to the period when it assumes an independent existence, embryology. Owing to the difficulty of obtaining material illustrating all the stages of this early development, gaps must be filled up by observations on the development of lower forms, comparative embryology, or by consideration of adult forms in the line of human ancestry, comparative anatomy. The direct application of the facts of human anatomy to the various pathological conditions which may occur constitutes the subject of applied anatomy. Finally, the appreciation of structures on or immediately underlying the surface of the body is frequently made the subject of special study, surface anatomy. Systematic anatomy. The various systems of which the human body is composed are grouped under the following headings. Osteology, the bony system or skeleton. Syndesmology, the articulations or joints. Myology, the muscles. With the description of the muscles, it is convenient to include that of the fascia which are so intimately connected with them. Angiology, the vascular system comprising the heart, blood vessels, lymphatic vessels, and lymph glands. Neurology, the nervous system. The organs of sense may be included in this system. Splanchnology, the visceral system. Topographically, the viscera form two groups, that is, the thoracic viscera and the abdomino-pelvic viscera. The heart, a thoracic viscus, is best considered with the vascular system. The rest of the viscera may be grouped according to their functions: A, the respiratory apparatus; B, the digestive apparatus; and C, the urogenital apparatus. Strictly speaking, the third subgroup should include only such components of the urogenital apparatus as are included within the abdomino-pelvic cavity, but it is convenient to study under this heading certain parts which lie in relation to the surface of the body, that is, the testes and the external organs of generation. For descriptive purposes, the body is supposed to be in the erect posture, with the arms hanging by the sides and the palms of the hands directed forward. The median plane is a vertical antero-posterior plane passing through the center of the trunk. This plane will pass approximately through the sagittal suture of the skull, and hence any plane parallel to it is termed a sagittal plane. A vertical plane at right angles to the median plane passes, roughly speaking, through the central part of the coronal suture, or through a line parallel to it. Such a plane is known as a frontal plane, or sometimes as a coronal plane. A plane at right angles to both the median and frontal planes is termed a transverse plane. The terms anterior or ventral, and posterior or dorsal, are employed to indicate the relation of parts to the front or back of the body or limbs, and the terms superior or cephalic, and inferior or caudal, to indicate the relative levels of different structures. Structures nearer to or farther from the median plane are referred to as medial or lateral, respectively. The terms superficial and deep are strictly confined to descriptions of the relative depth from the surface of the various structures. External and internal are reserved almost entirely for describing the walls of cavities or of hollow viscera. In the case of the limbs, the words proximal and distal refer to the relative distance from the attached end of the limb. End of section 0.
Section 1 of Gray's Anatomy, Part 1. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Ruth Golding. Anatomy of the Human Body, Part 1, by Henry Gray. Section 1. Embryology. The term embryology in its widest sense is applied to the various changes which take place during the growth of an animal from the egg to the adult condition. It is, however, usually restricted to the phenomena which occur before birth. Embryology may be studied from two aspects. One, that of ontogeny, which deals only with the development of the individual, and two, that of phylogeny, which concerns itself with the evolutionary history of the animal kingdom. In vertebrate animals, the development of a new being can only take place when a female germ cell or ovum has been fertilized by a male germ cell or spermatozoon. The ovum is a nucleated cell, and all the complicated changes by which the various tissues and organs of the body are formed from it after it has been fertilized are the result of two general processes, viz., segmentation and differentiation of cells. Thus, the fertilized ovum undergoes repeated segmentation into a number of cells which at first closely resemble one another, but are, sooner or later, differentiated into two groups: one, somatic cells, the function of which is to build up the various tissues of the body, and two, germinal cells, which become embedded in the sexual glands, the ovaries in the female and the testes in the male, and are destined for the perpetuation of the species. Having regard to the main purpose of this work, It is impossible in the space available in this section to describe fully or illustrate adequately all the phenomena which occur in the different stages of the development of the human body. Only the principal facts are given, and the student is referred for further details to one or other of the text books on human embryology. 1. The animal cell. All the tissues and organs of the body originate from a microscopic structure (the fertilized ovum), which consists of a soft jelly-like material enclosed in a membrane and containing a vesicle or small spherical body, inside which are one or more denser spots. This may be regarded as a complete cell. All the solid tissues consist largely of cells essentially similar to it in nature, but differing in external form. In the higher organisms, a cell may be defined as a nucleated mass of protoplasm of microscopic size. Its two essentials, therefore, are a soft jelly-like material similar to that found in the ovum, and usually styled cytoplasm, and a small spherical body embedded in it, and termed a nucleus. Some of the unicellular protozoa contain no nuclei, but granular particles which, like true nuclei, stain with basic dyes. The other constituents of the ovum, viz., its limiting membrane and the denser spot contained in the nucleus, called the nucleolus, are not essential to the type cell, and in fact many cells exist without them. Cytoplasm (protoplasm) is a material probably of variable constitution during life, but yielding on its disintegration bodies chiefly of proteid nature. Lecithin and cholesterin are constantly found in it, as well as inorganic salts, chief among which are the phosphates and chlorides of potassium, sodium, and calcium. It is of a semi-fluid, viscid consistence, and probably colloidal in nature. The living cytoplasm appears to consist of a homogeneous and structureless ground substance, in which are embedded granules of various types. The mitochondria are the most constant type of granule, and vary in form from granules to rods and threads. Their function is unknown. Some of the granules are proteid in nature and probably essential constituents. Others are fat, glycogen, or pigment granules, and are regarded as adventitious material taken in from without, and hence are styled cell inclusions or paraplasm. When, however, cells have been fixed by reagents, a fibrillar or granular appearance can often be made out under a high power of the microscope. The fibrils are usually arranged in a network or reticulum, to which the term spongioplasm is applied, the clear substance in the meshes being termed hyaloplasm. The size and shape of the meshes of the spongioplasm vary in different cells, and in different parts of the same cell. The relative amounts of spongioplasm and hyaloplasm also vary in different cells, the latter preponderating in the young cell, and the former increasing at the expense of the hyaloplasm as the cell grows. Such appearances in fixed cells are no indication whatsoever of the existence of similar structures in the living, although there must have been something in the living cell to give rise to the fixed structures. The peripheral layer of a cell is in all cases modified either by the formation of a definite cell membrane, as in the ovum, or more frequently in the case of animal cells by a transformation, probably chemical in nature, which is only recognizable by the fact that the surface of the cell behaves as a semi-permeable membrane. Nucleus. The nucleus is a minute body embedded in the protoplasm, and usually of a spherical or oval form, its size having little relation to that of the cell. It is surrounded by a well-defined wall, the nuclear membrane. This encloses the nuclear substance (nuclear matrix), which is composed of a homogeneous material in which is usually embedded one or two nucleoli. In fixed cells, the nucleus seems to consist of a clear substance or karyoplasm, and a network or karyomytome. The former is probably of the same nature as the hyaloplasm of the cell, but the latter, which forms also the wall of the nucleus, differs from the spongioplasm of the cell substance. It consists of fibres or filaments arranged in a reticular manner. These filaments are composed of a homogeneous material known as linin, which stains with acid dyes, and contains embedded in its substance particles which have a strong affinity for basic dyes. These basophil granules have been named chromatin or basichromatin, and owe their staining properties to the presence of nucleic acid. Within the nuclear matrix are one or more highly refracting bodies termed nucleoli, connected with the nuclear membrane by the nuclear filaments. They are regarded as being of two kinds. Some are mere local condensations (net knots) of the chromatin. These are irregular in shape and are termed pseudonucleoli. Others are distinct bodies differing from the pseudonucleoli both in nature and chemical composition. They may be termed true nucleoli and are usually found in resting cells. The true nucleoli are oxyphil, i.e. they stain with acid dyes. Most living cells contain, in addition to their protoplasm and nucleus, a small particle which usually lies near the nucleus and is termed the centrosome. In the middle of the centrosome is a minute body called the centriole, and surrounding this is a clear spherical mass known as the centrosphere. The protoplasm surrounding the centrosphere is frequently arranged in radiating fibrillar rows of granules, forming what is termed the attraction sphere. Reproduction of cells. Reproduction of cells is effected either by direct or by indirect division. In reproduction by direct division, the nucleus becomes constricted in its centre, assuming an hourglass shape, and then divides into two. This is followed by a cleavage or division of the whole protoplasmic mass of the cell, and thus two daughter cells are formed, each containing a nucleus. These daughter cells are at first smaller than the original mother cell, but they grow and the process may be repeated in them, so that multiplication may take place rapidly. Indirect division, or karyokinesis (karyomitosis), has been observed in all the tissues: generative cells, epithelial tissue, connective tissue, muscular tissue, and nerve tissue. It is possible that cell division may always take place by the indirect method. The process of indirect cell division is characterized by a series of complex changes in the nucleus, leading to its subdivision. This is followed by cleavage of the cell protoplasm. Starting with the nucleus in the quiescent or resting stage, these changes may be briefly grouped under the four following phases. 1. Prophase. The nuclear network of chromatin filaments assumes the form of a twisted skein or spirum, while the nuclear membrane and nucleolus disappear. The convoluted skein of chromatin divides into a definite number of V-shaped segments or chromosomes. The number of chromosomes varies in different animals, but is constant for all the cells in an animal of any given species. In man, the number is given by Fleming and Duesberg as 24. Coincidentally with or preceding these changes, the centriole, which usually lies by the side of the nucleus, undergoes subdivision, and the two resulting centrioles, each surrounded by a centrosphere, are seen to be connected by a spindle of delicate achromatic fibres, the achromatic spindle. The centrioles move away from each other, one toward either extremity of the nucleus, and the fibrils of the achromatic spindle are correspondingly lengthened. A line encircling the spindle midway between its extremities or poles is named the equator, and around this the V-shaped chromosomes arrange themselves in the form of a star, thus constituting the mother star or monaster. 2. Metaphase. Each V-shaped chromosome now undergoes longitudinal cleavage into two equal parts or daughter chromosomes, the cleavage commencing at the apex of the V and extending along its divergent limbs. 3. Anaphase. The daughter chromosomes, thus separated, travel in opposite directions along the fibrils of the achromatic spindle toward the centrioles, around which they group themselves, and thus two star-like figures are formed, one at either pole of the achromatic spindle. This constitutes the diaster. The daughter chromosomes now arrange themselves into a skein or spirum, and eventually form the network of chromatin which is characteristic of the resting nucleus. 4. Telophase. The cell protoplasm begins to appear constricted around the equator of the achromatic spindle, where double rows of granules are also sometimes seen. The constriction deepens, and the original cell gradually becomes divided into two new cells, each with its own nucleus and centrosome, which assume the ordinary positions occupied by such structures in the resting stage. The nuclear membrane and nucleolus are also differentiated during this phase. End of section 1. Recording by Ruth Golding. Section 2 of Gray's Anatomy, Part 1. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Ruth Golding. Anatomy of the Human Body, Part 1, by Henry Gray. Embryology. 2. The ovum. The ova are developed from the primitive germ cells which are embedded in the substance of the ovaries. Each primitive germ cell gives rise, by repeated divisions, to a number of smaller cells termed oogonia, from which the ova or primary oocytes are developed. Human ova are extremely minute, measuring about 0.2 millimeters in diameter, and are enclosed within the egg follicles of the ovaries. As a rule, each follicle contains a single ovum, but sometimes two or more are present. Footnote. See description of the ovary on a future page. End of footnote. By the enlargement and subsequent rupture of a follicle at the surface of the ovary, an ovum is liberated and conveyed by the uterine tube to the cavity of the uterus. Unless it be fertilized, it undergoes no further development and is discharged from the uterus. But if fertilization take place, it is retained within the uterus and is developed into a new being. In appearance and structure, the ovum differs little from an ordinary cell, but distinctive names have been applied to its several parts. Thus, The cell substance is known as the yolk or ooplasm, the nucleus as the germinal vesicle, and the nucleolus as the germinal spot. The ovum is enclosed within a thick transparent envelope, the zona striata or zona pellucida, adhering to the outer surface of which are several layers of cells derived from those of the follicle, and collectively constituting the corona radiata. Yolk. The yolk comprises 1. The cytoplasm of the ordinary animal cell with its spongioplasm and hyaloplasm. This is frequently termed the formative yolk. 2. The nutritive yolk or deutoplasm, which consists of numerous rounded granules of fatty and albuminoid substances embedded in the cytoplasm. In the mammalian ovum, the nutritive yolk is extremely small in amount, and is of service in nourishing the embryo in the early stages of its development only, whereas in the egg of the bird there is sufficient to supply the chick with nutriment throughout the whole period of incubation. The nutritive yolk not only varies in amount, but in its mode of distribution within the egg. Thus, in some animals it is almost uniformly distributed throughout the cytoplasm. In some it is centrally placed and is surrounded by the cytoplasm. In others it is accumulated at the lower pole of the ovum, while the cytoplasm occupies the upper pole. A centrosome and centriole are present, and lie in the immediate neighbourhood of the nucleus. Germinal vesicle. The germinal vesicle, or nucleus, is a large spherical body which at first occupies a nearly central position, but becomes eccentric as the growth of the ovum proceeds. Its structure is that of an ordinary cell nucleus, viz., it consists of a reticulum or karyomitome, the meshes of which are filled with karyoplasm, while connected with, or embedded in, the reticulum are a number of chromatin masses or chromosomes, which may present the appearance of a skein, or may assume the form of rods or loops. The nucleus is enclosed by a delicate nuclear membrane, and contains in its interior a well-defined nucleolus or germinal spot. Coverings of the ovum. The zona striata or zona pellucida is a thick membrane which, under the higher powers of the microscope, is seen to be radially striated. It persists for some time after fertilization has occurred, and may serve for protection during the earlier stages of segmentation. It is not yet determined whether the zona striata is a product of the cytoplasm of the ovum, or of the cells of the corona radiata, or both. The corona radiata consists of 2 or 3 strata of cells. They are derived from the cells of the follicle, and adhere to the outer surface of the zona striata when the ovum is set free from the follicle. The cells are radially arranged around the zona, those of the innermost layer being columnar in shape. The cells of the corona radiata soon disappear. In some animals they secrete, or are replaced by, a layer of adhesive protein, which may assist in protecting and nourishing the ovum. The phenomena attending the discharge of the ova from the follicles belong more to the ordinary functions of the ovary than to the general subject of embryology, and are therefore described with the anatomy of the ovaries. Footnote: see description of the ovary on a future page. End of footnote. Maturation of the ovum. Before an ovum can be fertilized, it must undergo a process of maturation or ripening. This takes place previous to or immediately after its escape from the follicle, and consists essentially of an unequal subdivision of the ovum, first into 2 and then into 4 cells. 3 of the 4 cells are small, incapable of further development, and are termed polar bodies or polarcytes, while the 4th is large and constitutes the mature ovum. The process of maturation has not been observed in the human ovum, but has been carefully studied in the ova of some of the lower animals, to which the following description applies. It was pointed out on page 37 that the number of chromosomes found in the nucleus is constant for all the cells in an animal of any given species, and that in man the number is probably 24. This applies not only to the somatic cells, but to the primitive ova and their descendants. For the purpose of illustrating the process of maturation, a species may be taken in which the number of nuclear chromosomes is 4. If an ovum from such be observed at the beginning of the maturation process, it will be seen that the number of its chromosomes is apparently reduced to 2. In reality, however, the number is doubled, since each chromosome consists of 4 granules grouped to form a tetrad. During the metaphase, see page 37, each tetrad divides into 2 dyads, which are equally distributed between the nuclei of the 2 cells formed by the first division of the ovum. One of the cells is almost as large as the original ovum, and is named the secondary oocyte. The other is small, and is termed the first polar body. The secondary oocyte now undergoes subdivision, during which each dyad divides and contributes a single chromosome to the nucleus of each of the 2 resulting cells. This second division is also unequal, producing a large cell, which constitutes the mature ovum, and a small cell, the second polar body. The first polar body frequently divides while the second is being formed, and as a final result 4 cells are produced, viz., the mature ovum and 3 polar bodies, each of which contains 2 chromosomes, i.e., 1/2 the number present in the nuclei of the somatic cells of members of the same species. The nucleus of the mature ovum is termed the female pronucleus. End of section 2. Recording by Ruth Golding. Section 3 of Gray's Anatomy, Part 1. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Ruth Golding. Anatomy of the Human Body, Part 1, by Henry Gray. Embryology. 3. The spermatozoan. The spermatozoa or male germ cells are developed in the testes and are present in enormous numbers in the seminal fluid. Each consists of a small but greatly modified cell. The human spermatozoon possesses a head, a neck, a connecting piece or body, and a tail. The head is oval or elliptical but flattened, so that when viewed in profile it is pear-shaped. Its anterior 2/3 are covered by a layer of modified protoplasm, which is named the head cap. This in some animals, e.g. the salamander, is prolonged into a barbed spear-like process or perforator, which probably facilitates the entrance of the spermatozoon into the ovum. The posterior part of the head exhibits an affinity for certain reagents and presents a transversely striated appearance, being crossed by 3 or 4 dark bands. In some animals, a central rod-like filament extends forward for about 2/3 of the length of the head, while in others a rounded body is seen near its centre. The head contains a mass of chromatin, and is generally regarded as the nucleus of the cell surrounded by a thin envelope. The neck is less constricted in the human spermatozoon than in those of some of the lower animals. The anterior centriole, represented by 2 or 3 rounded particles, is situated at the junction of the head and neck, and behind it is a band of homogeneous substance. The connecting piece or body is rod-like and is limited behind by a terminal disc. The posterior centriole is placed at the junction of the body and neck, and, like the anterior, consists of 2 or 3 rounded particles. From this centriole, an axial filament, surrounded by a sheath, runs backward through the body and tail. In the body, the sheath of the axial filament is encircled by a spiral thread, around which is an envelope containing mitochondria granules, and termed the mitochondria sheath. The tail is of great length, and consists of the axial thread or filament surrounded by its sheath, which may contain a spiral thread or may present a striated appearance. The terminal portion or end piece of the tail consists of the axial filament only. Krause gives the length of the human spermatozoon as between 52 μ and 62 μ, the head measuring 4 to 5 μ, the connecting piece 6 μ, and the tail from 41 μ to 52 μ. By virtue of their tails, which act as propellers, the spermatozoa are capable of free movement, and if placed in favourable surroundings, e.g. in the female passages, will retain their vitality and power of fertilizing for several days. In certain animals, e.g. bats, it has been proved that spermatozoa retained in the female passages for several months are capable of fertilizing. The spermatozoa are developed from the primitive germ cells which have become embedded in the testes, and the stages of their development are very similar to those of the maturation of the ovum. The primary germ cells undergo division and produce a number of cells termed spermatogonia, and from these the primary spermatocytes are derived. Each primary spermatocyte divides into 2 secondary spermatocytes, and each secondary spermatocyte into 2 spermatids or young spermatozoa. From this it will be seen that a primary spermatocyte gives rise to 4 spermatozoa. On comparing this process with that of the maturation of the ovum, it will be observed that the primary spermatocyte gives rise to 2 cells, the secondary spermatocytes, and the primary oocyte to 2 cells, the secondary oocyte and the first polar body. Again, the 2 secondary spermatocytes, by their subdivision, give origin to 4 spermatozoa, and the secondary oocyte and first polar body to 4 cells, the mature ovum, and 3 polar bodies. In the development of the spermatozoa, as in the maturation of the ovum, there is a reduction of the nuclear chromosomes to 1/2 of those present in the primary spermatocyte. But here the similarity ends, for it must be noted that the 4 spermatozoa are of equal size, and each is capable of fertilizing a mature ovum, whereas the 3 polar bodies are not only very much smaller than the mature ovum, but are incapable of further development, and may be regarded as abortive ova. 4. Fertilization of the ovum. Fertilization consists in the union of the spermatozoon with the mature ovum. Nothing is known regarding the fertilization of the human ovum, but the various stages of the process have been studied in other mammals, and from the knowledge so obtained, it is believed that fertilization of the human ovum takes place in the lateral or ampullary part of the uterine tube, and the ovum is then conveyed along the tube to the cavity of the uterus, a journey probably occupying 7 or 8 days, and during which the ovum loses its corona radiata and zona striata, and undergoes segmentation. Sometimes the fertilized ovum is arrested in the uterine tube and there undergoes development, giving rise to a tubal pregnancy. Or it may fall into the abdominal cavity and produce an abdominal pregnancy. Occasionally the ovum is not expelled from the follicle when the latter ruptures, but is fertilized within the follicle and produces what is known as an ovarian pregnancy. Under normal conditions, only one spermatozoon enters the yolk and takes part in the process of fertilization. At the point where the spermatozoon is about to pierce, the yolk is drawn out into a conical elevation termed the cone of attraction. As soon as the spermatozoon has entered the yolk, the peripheral portion of the latter is transformed into a membrane, the vitelline membrane, which prevents the passage of additional spermatozoa. Occasionally a second spermatozoon may enter the yolk, thus giving rise to a condition of polyspermy. When this occurs, the ovum usually develops in an abnormal manner and gives rise to a monstrosity. Having pierced the yolk, the spermatozoon loses its tail, while its head and connecting piece assume the form of a nucleus containing a cluster of chromosomes. This constitutes the male pronucleus, and associated with it there are a centriole and centrosome. The male pronucleus passes more deeply into the yolk, and coincidentally with this, the granules of the cytoplasm surrounding it become radially arranged. The male and female pronuclei migrate toward each other, and, meeting near the centre of the yolk, fuse to form a new nucleus, the segmentation nucleus, which therefore contains both male and female nuclear substance. The former transmits the individualities of the male ancestors, the latter those of the female ancestors, to the future embryo. By the union of the male and female pronuclei, the number of chromosomes is restored to that which is present in the nuclei of the somatic cells. End of section 3. Recording by Ruth Golding.